I'll give you the outline. We'll talk about antimicrobial resistance. Here we probably talk about what do we know and why do we care about this. We look into role of diagnostic lab into the whole program of antimicrobial stewardship. And I'll talk about some of the issues we see with how we're doing the work right now. These are the traditional methods we use in diagnostic bacteriology, and look at, what can be like, what are the solutions, what we are thinking. And we'll talk a little bit about some of the application we do here at PDS and what are the goals, what do we see in the future, what are our dreams. But before I do that, I would like to introduce ourselves. I don't know if everybody is familiar with Prairie Diagnostic Services. This is the formerly the Veterinary Diagnostic Laboratory at the Western College of Veterinary Medicine. And it has been around for many years as far as the Western College exists. So in 1998, the government decided to incorporate that into a non-profit corporation, and it's owned by the University of Saskatchewan and the Ministry of Agriculture. And when we started, we had to, they combined these two labs. We had the lab here at the Vet College and the lab into the lab in Regina. That's the provincial veterinary lab. And this lasted for almost 10 years when the government decided again to put all the effort and just focus in one location, that is Saskatoon, at the college here. So since then, we are offering different tests, close to 600. We have many clients. We serve all the veterinarian, the clinics in the province. We serve also some of the clinics in Manitoba, in Alberta. Some of you know Alberta doesn't have a diagnostic lab, veterinary diagnostic lab. So we have a lot of clients from Alberta and, of course, British Columbia as well. As services, we offer basically a full veterinary diagnostic services, and we have anatomic pathology, where we have the necropsy service, necropsy suite. We offer histology, we offer dermatopathology, and then we have clinical pathology, where we offer the chemistry analysis. We have hematology, urinalysis, cytology, and we have the microbiology with all the ologies, where we have bacteriology, and this is where I am, bacteriology and mycology. We have other diagnosticians taking care of immunology, serology, virology. We do also parasitology in that same suite, and molecular diagnostic using all the PCR technology. And of course, we have the toxicology lab that was kind of left alone, and now we are housing them into the same lab. This is just to give you a picture of what it looks like. This is the microbiology lab, some pictures, and with different stations. So all the services I just mentioned of in the microbiology, they are in the same big lab. And what you see here is just some of the section on the bacteriology. As I do bacteriology, we have some sections where people receive their sample and they're working on the 24 hours after incubation. This is 24-hour reading, 48-hour reading. You have some station where you have your microscope, an aerobic chamber, and all the different equipment. We'll talk about Malditov. This is about 3 or 4 years now. No, 3 years. We have that equipment. Have just changed the way we do bacteriology, and some other stations where we do some other testing, especially the salmonella. We are helping the Saskatchewan egg producers checking at least some fluff for salmonella presence. So this is who we are and where we are. So we are part of a system here where we give service to all these companion animals and all exotic livestock. So some of us, we do also participate in teaching at the college here, and we provide services to the ministry by providing all the surveillance data. And as I said, we also do some testing from other provinces. So what we are trying to do is just to make our animals healthy so that we can have healthy people. And of course, you don't spread all the disease in the environment and healthy environment. Now, so you can see that we are part, and we would like to be part of the whole system where we provide the service. We know we provide the service, but we just want to be part of a system where, you know, you can utilize new technologies, and you can partner with researchers and provide some education and transmission of knowledge to our clients. Now, antimicrobial resistance, we know, is a big issue, and right now it's a priority. It becomes priority number one for everybody. Recognized by everybody, basically, and we know how it affects humans, how it affects animals, and have a very negative impact on our health. There are some, not to be alarmist, but it seems if we don't do anything right now, you know, in about 30 years, then we'll have even a biggest problem of antimicrobial. It's just, for now, you can just, when you go to hospital, you see people with infection. They were coming down with multi-drug resistant bacteria, because we are talking about this bacteria. And we really appreciate that, and we have to be able to connect with everybody and to be able to work and do something about this. So it's everybody should be involved in this, and it's high priority. Now, as we know, there was very high level of political endorsement, as provided from some document from the Transatlantic Task Force on Antimicrobial Resistance. We have a very high-level meeting at United Nations General Assembly in September 2016. And even here in Canada, you have the House of Commons Standing Committee on Health. And that came, it's a big document that came last year. Not only on the politics, but you look at this scientific meetings as well. Last year there was a good meeting in Sorrento, Italy, with World Organization for Animal Health. There was a session only for antimicrobial resistance, and they talk about this and how we can help by coming up with rapid diagnosis. So we certainly know what is happening, and we care. And if we care, we have to think about what can we do to help out. As we know, antimicrobial resistance is really a quite complex topic, but at least on the veterinary medicine side, we know that inappropriate antimicrobial use, it's just one of the cause of the problem. It's not only in veterinary medicine, even in human medicine. The way we use antibiotics, maybe we need to rethink about it and try to do something. You know, we are not at the level where we're kind of blaming each other, who's responsible for this and so on. But now we know that, you know, it makes sense just to come together and general consensus that we need to do something to probably change the way we use antibiotics to treat humans and to treat our animals. There were some recommendations to fight antimicrobial. World Health Organization Global Action Plan, Pan-Canadian Antimicrobial Framework. So these people, they are kind of challenging all of us, scientists, diagnosticians, whoever, try to provide support to the program because we are coming with antimicrobial stewardship program. How can we help? At the National Stewardship Initiative here in Canada, you know, they're coming with some changes, and that will be a big change by December 1st this year. We are not going to use antimicrobial in veterinary medicine as it used to be. You need to have a prescription, and that will be a big change in veterinary medicine. So it's coming pretty soon, and yeah, things are happening, and see how we can help with that. So now all these, we come with, as I said, that the antimicrobial stewardship program promote careful usage of antimicrobial. And this, I think it's the role of all of us, all healthcare providers. So be it in agriculture, in public health, environment, food safety, we need to do something through education where we can come up with guidelines, regulation, how to use the drugs, what are the right dose, the right route of administration, the right duration, and so on. And of course, in veterinary medicine, try to justify metaphylaxis. We can also help by infection prevention and control, surveillance program to come up with some early warning system where we can detect antimicrobial resistant bacteria, and we can come up with some good ideas, innovation, how we want to deliver the service, because we've been doing, at least in the diagnostic lab, for so many years, the way we are providing the service. And what I can remember is you probably spend a lot of time trying to identify these organisms and come up with antimicrobial. I'm going to talk a little bit in details about this. And that's probably not the way we should provide the service now. We have to rethink the whole service providing into the diagnostic lab. So we have to rethink the way we do that, revisit our classical bacteriology delivery. We need to explore some other technologies and how we relate the information from the bacteriology lab to our clients, to our veterinarian, or to our physicians. So when we go into the conventional technology, you certainly recognize this. This is just a simple example where you are suspecting urinary tract infection, and you get your sample to the bacteriology lab and then culture it. So just to get the sample to the lab, sometimes it takes 1 day. And by the time it gets to the lab, you need to culture it, and you need to know what organism is involved in that by using some basic routine media or selective or some enrichment media. Here I'm giving just an example of urine, but can be some other body fluids where you need to do some enrichment and things like that. So with that, then you have to look at the microscope. You need to do some biochemical tests. And I remember bacteriologists used to spend a lot of time just trying to identify what organisms are involved in a situation, in a condition. And of course, you are a good bacteriologist when you can put a name on some organism. And it didn't matter how long it takes you to tell if the organism is important or not, but when you get the right identification, you are very good. So you go into all these systems before you come up with a name. So I think this is the problem with the conventional method. And as I mentioned, you have long turnaround time. By the time you get your sample, and by the time you get an answer, and by the time you get your antimicrobial susceptibility test, it takes too long. Although the World Organization for Animal Health suggests that you have to do all the testing, but it's the time to get the information to the attending veterinarian that is very too long and probably not acceptable. We can just think about a situation where you have a patient with an infection. In this case, I was giving you the example of urinary tract infection. What do? You don't want to wait 2 days or 3 days before you start treatment. You do something right away. That's a big issue. So maybe some of the solutions, okay, can you look into how we can deliver the information as quickly as possible? Here, again, bacterial identification and antibacterial. So for some years now, I think we've made progress. We need to recognize that because now we can provide bacterial identification within a day. At least we know what is involved in that. And some labs, they use Vitek 2 technology. Some of us, we use Malditov. And you provide identification very quickly. And antimicrobial susceptibility. I can say, at least in veterinary medicine, we've made some progress. I remember when I started, we had only one chart. And this is your interpretation for everything. And that was based on medical microbiology, medical bacteriology, because you can just think a dog is different from a cat, is different from a pig, different from a horse, different from a... So we are making progress, and these people from Clinical Laboratory Standard Institute, they are working very hard, and now we have different chart for interpretation for the different animal from different infections. So I think we are making progress. At least regionally, some of the lab can provide an antibiogram so that when you have an infection, you suspect an infection, and you know at least what is the agent involved in that infection, you can probably start something meaningful rather than trying different drugs because you have an antibiogram and you know what are the first line of antibiotics you can use for certain bacteria. Or we can also, to speed up the process, now we are in the era where we are looking into the whole genome sequencing. And, we have all the different platforms, the next generation sequencing platform, they are out there. And these are very good technology. Some labs, they have it. But we have to evaluate this into a clinical situation, see if we can use those. Just a picture of the Malditov, as I said. This has helped us a lot. And we don't have to worry about all these biochemical tests, only a few cases. But I believe like 90%, even more, we don't have to worry about bacterial identification, just making our life very easy. As I said, bacteriologists were spending probably half of the time reading and studying all these biochemical reaction and how you can identify organisms. So we've made progress. Antimicrobial, this is, again, something we still need to think about. What we do right now in our lab, we offer the Kirby-Bauer. So this is the disc diffusion. And we offer also the microdilution where you can provide the MICs. But the situation didn't change here. Still, you need, after identification of your organism, then you need another day to do the antimicrobial. So by the time you receive the sample to information to the veterinarian takes, in that situation about 2 to 3 days. So here I'm showing just the situation. You can appreciate what can happen in the field where you have animals dying, and by the time you get a veterinarian, attending veterinarian, okay, go at the in the field, go to the farm and look at what's happening, take a sample, and then try to send the sample to the lab. As you know, here in Saskatchewan, we don't have buses anymore. And I just heard that Greyhound is closing, not giving any service, Western Canada. So it just become complicated, transportation. And then by the time it gets to the lab, culture it, look at the microscope, do sensitivity, gather all the information, send to the veterinarian. So you're looking at 3 to even a week. If it's quite easy, like urine, I mentioned that, and if it's quite complex situation, you are looking at 1 week. There were some worst case scenario where you're looking even more than a week. So I think turnaround time, that's the problem. Now, what is the solution? Now this is something we probably need to start exploring. And we have all the technology, next generation sequencing. So it's quite interesting. And a lot of research lab, they look into whole genome sequencing for bacteria, for viruses, for parasites, insect, and so on. At least they can gather the information. Every time you have new technology, you need to know how it's working and how you can get the better information from this technology and provide that to your client. So I think with all these equipment, we are in a situation, we are in the era where we are exploring all these and see what can be better. And then there was the nanopore technology with MinION. As you can see, very small device, probably for clinical application. That can actually, you can dream about using this device. And I heard they use this device for the Ebola outbreak in Africa, where they were able to load their sample right into the device and do the sequencing and provide that equipment. So provide information needed. Maybe this is something we can probably think about it into our system. So they are coming with different devices where just trying to make life easier, where you don't have to worry about your DNA extraction and library preparation. So that can be done with using some equipment here, Voltrax. I understand now they're coming with new generation of, it's going so quickly, and we have to adapt. I think they are just trying to improve what we can get from all these technology. So after extraction, then you use the sequencing, and then, of course, you need to analyze your data just to make sure you're getting the information you want about all these bacteria looking into what are the virulence factors, what are the, well, started with identification for sure, and then look into all these virulence factors. And for us, what is interesting, look into the antimicrobial resistant genes. So this is quite promising. And what the dream, here is the dream is, as I just mentioned, by using this device, then you can certainly reduce your turnaround time. It's really fast. So you can think about an attending veterinarian in the field or a clinician in a veterinary clinic. Take your sample, let's so be it like a urine sample, load it on the device, and send it to a center for interpretation of data and get the result the same day. Is it realistic? Probably. This is what's happened, sorry. So what we're thinking, you have your reference lab where you can handle all the information, and then you have all the veterinarian clinics, and so these are your partner in the field. So they know you provide them with protocols, how they can handle the sample, how they can load the sample, and send back all the information to the diagnostic lab, which is a reference center. And these people then can translate these back to the veterinarian or the clinician within the day. So to do that, I think we need to build up teamwork to make those decisions. So, you know, you need your extension veterinarian, you need the microbiologists, epidemiologists, molecular biology, bioinformatics. You need just to get all these people together and then try to help each other, try to help with all the program antimicrobial stewardship. So what I'm thinking is, if you can do that, I mean, identity the same day and provide that information about at least antimicrobial resistant profile the same day, and then the physician or the veterinarian in the field knows exactly what they need to use and what not to use, that can be really helpful. Now, here at PDS, this is exactly what we are trying to do. And as I said, this needs, like this, you need to build a team where you have everybody involved in that. And, you know, if there are certain people who are quite interested in what we do, you know, we are open. We are open for suggestion. We are open for business, to say. So some of the application here, actually, we had we were able to attract some funding here. We are exploring the nanopore sequencing for rapid diagnosis of bovine respiratory disease. And this is a huge problem in veterinary medicine. We have another project where we look into practices. And I remember, Jason, when we were down to Regina, I was thinking about that. You had a very good project on urinary tract infection and see what are the practices to serve that. So I was doing something similar, and I was so interested in that. And I'll talk in two details about this. So this is the first program where we are looking at how we can get a sample from an animal to the lab and, you know, being like a lab of reference and give the information right away. So you know exactly what is in sample and you know what is the antimicrobial profile of whatever organism is present. So what we are doing right now, we are kind of running a pilot project where we collect the samples and we do just the routine bacterial culture as usual. We culture it, and in the meantime, we freeze the sample and we can explore the whole genome sequencing. So you have your sample, and then, of course, identification. And these are the pathogen involved in bovine respiratory diseases, Monohamia hemolytica, Pasteurella metosida, and the Staphylococcus somni. When you have these, then you run your MICs, and what we do right now, it's we take that same sample and then run it through whole genome sequencing and try to get information about identity and about antimicrobial resistance. So this is just preliminary data, and what I'm showing here is we have selected about 30 Monohamia isolate with different antimicrobial profile. As you can see, we are looking at a group of antimicrobial. We have beta-lactams, we have tetracycline. As you can see, we have some resistance here to tetracycline. They are quite susceptible to fluoroquinolone and phenicol, yeah, and some resistance aminoglucoside and macrolide. So we look into that, and then we try to compare these with whole genome sequencing. Can we have similar results when looking at whole genome sequencing? So what we did that, again, very preliminary data. We didn't, as you can remember, you know, we didn't have any resistance to beta-lactam. This is the same stuff we got from whole genome sequencing. So no resistance to beta-lactam to fluoroquinolone. And we had some resistance to tetracycline, aminoglycoside, and we are still analyzing. And when you have this data, you look at different database. And what we did so far, we look at the database, CARD database, but there are some other database there, and we are exploring this. We are looking just to see what we can get. So that's one of the application here, and we'll see in the future how we can push this. Basically, it's just to get from the sample and get the information. That's the whole idea. The other part is a kind of education. I'm just giving you an example here from the practices. So what our clients do, veterinarian, sometimes they can look at the animal and just by the behavior and probably suspect urinary tract infection. And in the clinic, what they do, they will do a SEDI stain. It's just a microscopic examination. They look what can be present, and just base a diagnostic, just make a diagnosis based on that. So in this case, this is a dog that was treated, and then say, okay, we know the dog had a urinary tract infection because they saw some cocci, white blood cells in the urine sample, treated with Convenia, and then, they didn't improve. After 9 days, well, symptoms are still there, and try again another SEDI stain. Well, cocci are still present, white blood cells still present. Okay, you know what? We'll send home with a different antimicrobial. So you can see the practice. Well, at this time, they decide to send the sample to the bacteriology lab. Say, well, we're probably dealing with resistant bacteria, or, it's, we are not using the right drug. So when we look at here, it was negative. And, we see these quite often. And last year, I did actually a study where I think over 2015, 16, I was just looking at the information I was getting to the bacteriology lab and try to capture these cases. And we came up with something. When urinalysis is done in the laboratory versus our culture, about 97% concordance between our lab and, but when you look at what's happened in the clinic, when they suspect, we just have about 44%. So you can imagine that in about 56% of cases, these people are just using unnecessary treatment. They're using antibiotics where they shouldn't. Now, this is based on this. Now we had a kind of summer project where we say, we'll collect urine sample from veterinary clinics, and they just go with their protocol. they based on the information with CD-stain and whatever, they can decide if they think it's urinary tract infection or not, and then what treatment. For us, we'll collect that urine sample, and then we'll run the culture and then urinalysis in the lab, and we'll provide finding and recommendation. At least at this time, I know we publish those observations in a newsletter, but we didn't see any change. Maybe this time, by providing this, maybe some scientific evidence or some sort of scientific publication, maybe we can provide that piece of education to our clients. So again, this is just one of the education piece we are kind of providing here to help with antimicrobial stewardship. So to summarize what we are trying to do here, our mission is to help with the antimicrobial stewardship program, help reduce turnaround time, help provide some rapid diagnosis. Of course, we'll still do traditional work, but we are exploring other technology, as I had mentioned, to be able to provide this information rather than waiting 3-4 days. If we can provide that information within a day, that will be our dream come true. So this is what we wanted to share with you today. This is our people in the lab, happy faces, working very hard. I hope you enjoyed the presentation, and if there are some questions, we'll be able to address those. I didn't mention that we have Kazal here, we have Anatoly, and we have Igor. Okay, Igor just joined us today for a visit. So thank you very much.
Thank you, Ms. Angu. That was excellent. This is Jason and Regina here. I guess, first of all, I just want to say thank you guys for taking the time to do this presentation. I'm always fascinated to hear what's going on with you guys, and I think every time we chat or I listen to you guys whenever you have something to say, it reinforces for me the sort of parallel tracks that our worlds are on, and speaking animal world and human world, I guess, where you talk about some of the hurdles you guys are up against in terms of performing diagnostics in a timely manner and getting the information back in a timely manner. It just reminds me, we go out and we talk to family physicians, and one of their biggest concerns is, I need point of care testing so I can help determine is this a viral illness or a bacterial illness, to stop some of that even initial treatment that may be unnecessary. And then certainly when I think about our hospital situation, we just recently had a PCR system set up here so we can do PCR analysis for blood infections where they suspect. So if you have Staph aureus, whether it's methicillin resistant or methicillin sensitive, so getting a much quicker turnaround within hours rather than days to look at whether you have MRSA or MSSA. And so that is helping to eliminate some of the, or helping to focus that antimicrobial therapy much quicker and eliminate unnecessary use where that is happening. So anyways, I guess just to say, I think it's fantastic, and I don't know what other people from the human health world and the human laboratory world have been able to tune in for this, but I just think it's great to see some of those parallels. And the other piece I just wanted to say too was, I think that I love that the canine urinary tract infection project. I think that's very cool, and it's a great way to start looking at some of that appropriateness and feeding that information back to the clinics, just so they have an idea about what this looks like. Absolutely.
Mm-hmm. No, thank you for the comment. Anything to add here? Okay, just a second.
Hi, yes. So yes, this is really fascinating technology. I can see a lot of useful applications in the clinical field. My question was in regards to cost. So from a veterinary perspective, a lot of the time clients will refuse to do culture and sensitivity because it's too expensive. So when do you project it'll be cost effective?
For now, I think we don't have to worry about the cost. With all this technology, if you remember, just to do the whole genome sequencing, how much was it? $1 million or something? I don't remember. But now, we are going into like 1,000, even lower than that. And I don't think we need to worry about the cost. It will eventually come down and most of the time people, I think, with all the new technology, they keep that in mind. They know the cost is a big issue, and they always try to go lower and lower. If you look at this device, what was it the cost again when they started? It was 1,000, and now they're talking about providing this for 500 or even less, something, less than 100. So yes, of course, when you start, the cost will be an issue, but as we move along, hey, for example, now I do a lot of sequencing in the lab. In the past, we were doing this sequencing for $250. Now is about $50, and now in South Korea, they do sequencing for about $10. We send all our sample to South Korea, $10. And everybody is kind of, oh yeah, please go with sequencing. So yeah, I'm quite confident with all these costs will certainly go down one day. Any other comments, Anatoly?
I definitely agree that cost is a big consideration both for human health and animal health. At the same time, I think we really need to be more concerned about building our competencies in areas that we haven't touched before. So, for example, bioinformatics is a huge field, and it looks like everything goes whole genome sequencing, and bioinformatics is still not our core competency. So, for us, I don't really want to commit to any specific date or price tag when this service will go live, but I really want to say that it's a useful exercise to learn, to explore, and to move forward, because that's where the future is going to. So any other questions or comments or suggestions? I see people are starting slowly disappearing from the screen. It's a busy day, so one more time, I would like to thank you very much for this presentation, Musengo. It was a great overview, and thank you so much, everyone who was able to join us for this lunch hour presentation.